Now, one of the important concepts about enzymes to grasp is that they are highly specific. They are specific molecules designed for a specific purpose. And specifically, that they are specific to the substrate that they're going to act upon, the temperature at which they are going to act, and the pH of the environment in which they are acting. So first of all, let's think about the nature of the substrate. Now here we have our enzyme with its highly specific molecular architecture. And here we have the active sites. These are the active sites. And we'll suppose that this is the sucrase enzyme we have been considering. Now, here's a molecule coming along, and this molecule might be, for example, an amino acid. And because of kinetic theory, we've got movement of the enzyme in the fluids and movement of the amino acids, two amino acids here, small protein, part of a polypeptide. And they're moving around, and they might just happen to come together like that. Now, can we see that there's no reciprocal shape between the substrate, or the potential substrate, and the enzyme here? So that's just going to wander off into space again, because there's going to be no bonding, there's going to be no substrate enzyme complex formed. There's going to be no temporary bonds because everything's in the wrong shape. All the electrochemical components of the enzyme and the substrate don't match up, and the physical shape doesn't match up. The whole thing's completely hopeless and doomed from the start, so that will just wander off into space like that. But let's suppose the correct substrate comes along, in this case sucrose. That's now going to, again by random movement, it's going to sometimes fit into there. Oh, that looks a bit better, doesn't it? So now we've got a perfect match between the substrate, this is the sucrose molecule, and the sucrase enzyme. We now have a perfect match between these two. So we see that enzymes are specifics for substrate. That one simply won't do, but that one fits perfectly. So high specificity in terms of the substrate that can be enzymically catalyzed by the reaction by the enzyme. Now, enzymes are also highly specific to temperature. So let's see what's going on here in terms of specificity for temperature. Now, you're probably familiar with kinetic theory. Now, what kinetic theory does is it says any particle, it doesn't matter what it is. It could be my lens cap here as long as it's a microscopic particle. These microscopic particles are going to be acted on by molecules in the fluid. So in a fluid there's going to be lots of molecules, probably mostly water. Lots of molecules like this all around. And as the temperature increases, the energy of those molecules is going to increase. So if we take any one molecule, it's going to be vibrating more with increasing temperatures. And if we increase the temperatures more, the vibration is going to increase. So for any individual particle, let's suppose this pen lid is an individual water molecule, that's going to be vibrating at a rate which is determined by the temperature. That's what heat is, it's molecular vibration. So there it would be very cold because it's still. But as it gets warmer, it moves more, and as it gets hotter and hotter, it moves more. So heat is molecular vibration, that's what it is, and it occurs in a chaotic direction. But if we have larger particles here, like this, still a microscopic particle but larger, then that's going to be acted on by water molecules from that side, and from that side, and from that side, and from that side, and from that side. But remember, these water molecules are themselves acting in a chaotic way. So what will happen is, at any one moment in time, you might have several water molecules hitting it from that side, more than you have hitting it from that side. So what this means too is that these more macroscopic particles, not macroscopic but larger molecules, are also going to randomly vibrate. This is called Brownian motion. and it's caused by the vibration of the individual water molecules just hitting this in an irregular way, causing this movement. And it's the same because, of course, the substrate is going to be in a watery medium, so that's going to be vibrating, and as temperatures increase it's going to be vibrating more. And of course it's the same for the enzyme, that's going to be vibrating as well. So you can see if these are pretty still, there's not much chance they're going to come together. But if there's more vibrations, if there's more molecular vibration going on, then okay, sometimes it's not going to fit, but other times it's going to come in the right direction. So increasing temperature increases the probability of the substrate hitting the right active sites within the enzyme and forming the substrate enzyme complex. And then what we actually see, if we think about this graph, this explains what's happening here. So what we have here is we have enzymic activity there. So that's 100% when the enzyme's working as much as it possibly can, and that's 0 when it's not working at all. That's 50%. And here we have the temperature. So this is an enzyme which is working at physiological temperatures. So 0 degrees centigrade, 20, 40, 60 degrees centigrade. And we find out that as the molecular vibration increases with the kinetic theory and the Brownian motion of the substrate and the enzyme, that the probability of the substrate enzyme complex forming is going to increase as the temperature increases because there's more molecular vibration. And then it's going to reach a peak point here, and this is a physiological type enzyme, probably working in a body cell, that's reaching optimum at about 38 degrees centigrade. So that's good. But then if we carry on increasing the temperature, although it's true if we carry on increasing the temperature you're going to get even more Brownian type motion, but the trouble is, as we increase the temperature, because this active site is generated by very complex folding of the protein, what will happen if we increase the temperature further is we will get alteration of the physical shape of this and therefore the electrical chemical properties of this active site. It will physically change shape and chemically alter. And that is called denaturing of the active site. So they will be denaturing of the active site. So if we increase the temperature a little bit, we're going to get some denaturing of the active site. But then if we increase the temperature more, we get very rapid deterioration of the active site. And it becomes unrecognisable, therefore the shape of this is going to change completely, so it won't fit in. So with increasing temperatures we get denaturation, and that's irreversible. This protein is then damaged forever as a result of heat. In burn injuries we call this a coagulation necrosis. We get coagulation of the proteinaceous tissue and death of the proteinaceous tissue in a coagulation necrosis, which is what causes the burn injury essentially. It also happens when you fry an egg. When you put in the egg in your frying pan, it's the albumin, the white part is clear, but then it goes cloudy as the proteins denature. So we get denaturing of this and therefore rapid drop off in enzymic activity with increasing temperatures. So the enzyme is going to work optimally within a fairly narrow range of temperatures. Now the other specificity we mentioned about enzymes was pH. So we've talked about specificity for substrate temperature. Now we're thinking about the acidity or the alkalinity of the environment. So again we notice that this axis is referring to enzyme activity, 50% of activity, 100% of enzyme activity where it's working as quickly as it can. And here we have the pH along the bottom, so from a theoretical pH of 0 up to a neutral pH just there, and then a very alkaline pH up here at 14. And here we have an enzyme which is working at physiological pHs. You might remember the pH of arterial blood is 7.35 to 7.45. So this particular enzyme is working within that kind of range. But we notice that the range is relatively narrow. So the temperature graph actually was a bit spread out, wasn't it? There was potential activity between these 2 points. but with pH it's much more specific. This, of course, is why acidosis or alkalosis is such an important clinical finding. It's very serious if someone becomes acidotic or if they become alkalotic, because all of the body enzymes are no good, all of the somatic body enzymes, the intracellular enzymes, are nearly all attuned to work at these physiological pHs. And if that moves very much in either direction, either towards acidosis or towards alkalosis, then enzymic function in the cells will just not occur, and therefore there'll be no life-giving biochemistry. So quite a narrow range really for pH. And again, the rationale for this is much the same as it was for the increasing temperature. So at reduced pHs, the shape of the active site of the enzyme is going to be denatured. And it's the same with increased pH in alkaline environments. It's going to be denatured as well. So alkalosis or acidosis can cause denaturing of the active site of the enzyme, therefore meaning it's no longer the correct shape to accept its substrate. Therefore the enzyme will lose its ability to function in this physiological way, forming the substrate enzyme complex.